Hello everyone. Now we are going to see about innate and acquired immunity. This is immunology series where I will be teaching small lectures in immunology. So first, what is innate immunity? What are the cells that constitute innate immunity? You need to remember the word MNC, that is MNC, multinational company, which we commonly abbreviated as MNC. So macrophages, I make use of that. Macrophages, neutrophils, there is another N in our abbreviation. N is NK cells and the C is complement. So this is the basic for immunity. What does innate immunity is made up of? Macrophages, neutrophils, natural killer cells as well as the complement. They are the key players. The innate immunity, what happens is that they are germline coded receptors. What do they mean by germline coded receptors? It means the innate immune cells can detect the microbes right away using non-specific pre-coded receptors. But adaptive immunity takes some time. But this innate immunity, it has pre-coded receptors which are not specific. That is, they can be with these the receptors that are present on the innate immunity with the receptors. They can also recognize pathogen-associated molecular patterns, such as what generally in gram-negative bacteria we have lipopolysaccharide. In flagellin is also present on the bacteria. Viruses, they have double-stranded DNA. And in DNA and the viruses we have CpG DNA. So these are all pattern recognition. This pattern can be recognized by these receptors, that is the receptors which are present on the innate immunity. So the receptors are toll-like receptors or nod-like receptors. So in this way, that is, the receptors are not exactly specific, they are general to recognize the general patterns. So therefore, once it is recognized, it can produce and it can counteract the pathogen immediately. That is, lightning fast activation will happen within minutes to hours of the pathogen being exposed. But what is the problem over here? The response will be short-lived. Even though it has a lightning fast activation, the response will be short-lived because it is not catered to that specific pathogen. Instead, it is just general. So the germline coded receptors means it has been already coded in their DNA of the for which encodes the receptors to respond this and this way for this and this pathogen. The receptors are like that so that it can recognize the general pathogens. So this is the innate. This is about innate immunity. But on the other hand, we have acquired immunity. Acquired immunity, the special forces are T cells and the B cells, as well as the antibodies which are produced by the B cells. In contrary, here what happens is there is VDJ recognition. What do you mean by VDJ recombination or rearrangement? There are genes which are coding for the T cell as well as the B cell receptors, right? Those genes will undergo recombination. How? The genes will undergo a special cut as well as a paste process. So in that way, the receptor gene sequence is being changed so that the lymphocyte, that is either a T cell or a B cell, will end up with a unique receptor protein on its surface. That's what you should know. So once rearranged, that cell and all its descendants will make only that one type of receptor, that is a clonal specificity, which can fight against that particular pathogen. So it will be that much specific. So it will be precise for that pathogen which has entered the body. That is by means of this VDJ rearrangement, which happens at the gene level to rearrange and the gene has been rearranged such that that particular gene produces a one particular B cell receptor. So this is about the acquired immunity. But the problem is for the gene to get rearranged and for the receptor to get produced and then for the precise antibody to be formed or the precise receptor to be formed to combat the pathogen, it will take days to fully mobilize. There will be delayed activation. But we do have a positive aspect to it. There is long-term protection. The memory cells will provide rapid enhanced responses. Even on re-exposure to the same pathogen, there will be rapid response. The first time the response may be quite delayed because of the work that it has to do with respect to the gene rearrangement. But since it is produced, that rearrangement has happened, and that specific receptor is produced, and in addition to it, that clones of that particular lymphocyte or the B cells or the T cells have been produced in excess and is already present in the body. When the same pathogen reappears, when there is a re-exposure to the pathogen, then this can fight immediately and with the precision, and then we will be cleared of the infection. What about the natural killer cells? As I told you, the natural killer cells stands for one of the M in that MNC mnemonic, which are the innate immunity. But natural killer cell is one of the fascinating cells. In my previous video, I have told you about MHC class 1 and class 2. The MHC cells, they will take the peptides which are present inside the cell and it will expose outside. The peptides can be, just a summary, the peptides can be either the self peptides or it can be the peptides from the pathogen. So if it is a self, then the T cells and then the T cells are not stimulated and they will just patrol away. But if it is from the pathogen, then the T cells will kill the affected cell, right? If it is a cytotoxic T cell, that is CD8, where it will recognize the MHC class 1, it will kill the cell. But if you want in detail, you can see my previous video. But if it is an CD4 helper T cells, it has a different set of processes. But here, sometimes the viruses or the cancer cells are even more trickier or sinister. In a sinister way they behave. How? They will not allow the MHC to be expressed outside the cell, because only if the MHC is being expressed outside the cell, then the T cells which are patrolling outside in the blood will detect the MHC, right? That is, this MHC is holding this abnormal pathogen and hence that cell has to be killed. That's what the T cell is been programmed to do. So the viruses will want to play safe inside the cell, therefore it will not allow the MHC to express outside the cell. There comes the natural killer cells. The body is fascinating. How will the natural killer cells act? When it finds that the MHC is underexpressed in one particular cell, then it knows very well that it has been invaded by a virus or it has been invaded by a cancer. Therefore, the MHC army will kill that cell immediately when it notices an underexpression of the MHC. I mean, the natural killer cell army will now kill that invaded cell immediately, because it has found that the MHC is been underexpressed. Really fascinating, right? Yes, exactly. And you also need to know that the natural killer cells are activated by interleukin 12. No prior sensitization is needed, just under expression of the MHC is needed, and it is a rapid cytotoxic response. This is what I told you. When the MHC class 1 is downregulated to evade the immune detection, then natural killer cells recognize this missing self signal and it will initiate the targeted destruction. What about the antibody mediated dependent cell mediated cytotoxicity? The natural killer cells, B cells and T cells, represent the three lymphocyte types. Fine. There is something known as antibody binding. The plasma B cells will produce antibodies that will bind the antigens. The antibodies will have an FAB region as well as the FC region. The FAB region is always bound with the antigen. The FC region is present outward. The CD16, which is present on the NK cell, will bind to the exposed FC region of the bound antibodies. The multiple CD16 molecules will cross-link together just like this. The target cells are there. There are multiple CD16 which cross-link, triggering the natural killer cell activation as well as degranulation. Now the natural killer cells will release granzymes, granulins, perforins, and inducing the apoptosis of the target cell. So if this is the antibody, this is the FC, and this is the FAB. Here the antigen has been bound to the FAB, but here in the FC, the CD16 receptor of the natural killer cells are present. There will be multiple cross-linking of the CD16 of many natural killer cells, which will finally degranulate and kill the pathogen which is present on the FAB. This is antibody-dependent cell-mediated cytotoxicity. What do you know about B cells? The B cells are the main drivers of the humoral immunity. It will differentiate into plasma cells that will secrete massive quantities of antibodies. So what happens? The B cells first point is it can function as antigen presenting cells, phagocytosing the pathogens and presenting the antigens to CD4 plus T cells via MHC2. MHC2 is present on antigen presenting cells. So when the B cell which has phagocytosed a pathogen breaks it into smaller peptides and loads them onto MHC and presents it to the CD4 plus T cells. Now the CD4 plus T cells, on recognizing the pathogen, it will make the B cell to transform into a plasma cell such that it produces the specific antibody. For this transformation to happen, the B cell travels to the germinal center of the lymph nodes and then the transformation into plasma cells happen, and the germinal center of the lymph node along with the transformation into plasma cells, there is something known as somatic hypermutation which happens over there. What does somatic hypermutation mean? It undergoes genetic refinement to increase the antigenic antibody specificity. So it is actually going to produce an antibody, but it will be changing its genetic rearrangement to produce an antibody which is very much specific to the pathogen that has been attached, that has been phagocytosed initially. So this happens in the germinal centers, I told you. Now once that that is what is known as somatic hypermutation or tiny random changes happens, and that antibody with the small mutations happen such that it has such that it can bind with the antigen much more effectively. Then there is something known as affinity maturation. The immune system tests which antibody copies fit the antigen most tightly. So the B cells with better fitting antibodies get survival signals, and the B cells with weakly fitting antibodies will die off. So whichever antibody is attacking the antigen very perfectly, does the job very well, those B cells will survive and more of those antibodies are produced. Though those B cells which produced weakly fitting antibodies will die off by apoptosis. What is the clinical significance? Many leukemias and lymphomas will originate from the B cell lineages. Why? The B cell is supposed to undergo apoptosis. It is supposed to mature. Somatic hypermutation has to happen. Affinity maturation has to happen so that it caters to that particular antigen or the pathogen. But sometimes these regulations are missed and the B cells undergo uncontrolled proliferation resulting in leukemia and lymphoma. So this is the basis for leukemia and lymphoma arising from the B cells. With this, we have come to the end of lecture 2 in immunology. I'm happy. I will be happy if this helps you, and if you give me and if you show me that this has helped you by your thumbs up sign. And I have a good news to you. I'm starting my mentoring lessons for medical graduates as well as USMLE aspirants. I am a medical doctor. My name is Dr. Veena Velmurugan. I am an anesthesiologist. If you like my videos and if you want to get mentored by me, you can send me a message in the comments, and I'm looking forward to see you succeed. Have a good day.